Good afternoon, everyone. Are we live? I can't, it's hard for me to tell. Okay, I think we are live. So good afternoon, everyone. My name is Michelle Basso, and I'm delighted to welcome you to the 5th session of the NPRC's 60th Anniversary Scientific Symposium. Today's session focuses on precision medicine. Now, precision medicine represents a personalized or individualized approach to medicine, including prevention, diagnosis, and treatments. It takes into account individual variability in genes, environment, and lifestyle for each person. Modern precision medicine is driven by progress in biotechnology, revealing unprecedented details of individuals' molecular and genetic makeup. Precision medicine is aimed at advancing patient care by combining established and new therapeutic strategies targeted to individual needs. In this session, we will discuss the role of non-human primates, monkeys in particular, in supporting the development of precision medicine approaches, including emerging new models of genetic disorders, as well as personalized genome editing and cell therapies. As a reminder, please submit questions at any time through the Q&A function, and plan to remain on the line after the talks to hear answers to your questions and to participate in a live discussion with the speakers. So our first presentation is titled Combined Immunotherapy Using Mesenchymal Stem Cells for Reactivation and Chimeric Antigen Receptors, CAR T cells for targeting latent HIV. Our speaker is Stephen Braun, and Dr. Braun is an assistant professor of pharmacology at the Tulane National Primate Research Center. Take it away, Steve.
Hello, my name is Steve Braun. I'm in the Division of Immunology at the Tulane National Primate Research Center. First off, let me thank the organizers for inviting me to present today our work on MSC reactivation and CAR T cell targeting. During our preliminary meetings, we discussed what is precision medicine. Precision medicine is envisioned as a personalized approach to prevent, diagnose, and treat patients. It is driven by progress in biotechnology and generates disease-relevant molecular data. It combines established and new therapeutic strategies targeted to the individual. In this session, we will discuss the role of non-human primates in supporting the development of personalized medicine, including emerging new models of human genetic disorders, personalized genome editing, gene and cell therapies. Non-human primates are an excellent model for stem and gene therapy research. They are similar in size, anatomy, physiology, biochemistry, and behavior. They have similar immune system, both in development and in adaptive and innate systems. Additionally, many human reagents cross-react with non-human primates. They are outbred but have known MHC alleles, and human viruses often cause human-like diseases in NHPs, where challenge experiments in humans would not be possible. Thus, non-human primates have been a translational bridge for stem and gene therapy applications. In discussing precision or personalized medicine for either stem or gene therapy, we start with the donor or the patient and isolate somatic cells for characterization. These cells could be used directly in clinical applications such as mesenchymal stem cells or hematopoietic stem cells or T cells, or somatic cells could be reprogrammed into induced pluripotent stem cells. Additionally, these cells could be genetically modified to augment their functional activity. Our laboratory has been focused on combining MSCs for reactivation with CAR T cells to enhance the cytolytic activity against latent HIV reservoirs. Our initial strategy was to link anti-HIV CAR immunosurveillance with CMV-specific immune responses. We hypothesized that genetically modified CMV-specific CAR T cells would specifically persist in vivo in a functional effector memory type phenotype based on their CMV specificity and provide anti-HIV immune responses to the tissue. So by linking the CMV specific immune response to the CAR T cell, we can enhance the activity of the CAR T cell to mimic the CMV specific immune response. Therefore, we use CMV specific peptide pools to IE1, IE2, and PP65 to stimulate CMV specific T cells for expansion and transduction. So these data show in cryopreserved cells, the CMV specific immune response is polyfunctional, expressing both TNF and interferon alpha. When these cells are expanded in vitro for 10 days and then re-stimulated with CMV specific with the peptide pools, we can see that there's a large increase in the frequency of these CMV specific T cells and that they're still polyfunctional. If we look further at the phenotypic characteristics of these cells, we can see that they're mostly CD8 positive T cells. They have an effector memory CD95 positive, CD28 negative phenotype, and their expression of PD-1 is reduced compared to unstimulated or CD3, CD28 bead stimulated T cells. So we stimulated and transduced CMV-specific T cells with the CD4 CAR2 vector and co-transduced with the MAC46 vector, or we used the CEA control vector to transduce these cells. And these just show schematic diagram of what the CAR structures look like. So these data show the transduction and expansion of the CMV-specific CAR T cells. We took 20 million PBMCs and stimulated with the peptide pools, and on day 3, we transduced with the retroviral vectors. And then these data show the expansion of these CMV-specific cells over the course of 2 weeks. When we analyzed expression by flow, we can see that most of the cells are CD3 positive T cells, CD8 positive, and roughly 50% of the cells are expressing the CAR antigen. So, to mimic a clinical scenario, we challenged animals with the CCR5 tropic SHIV-D from George Shaw. We administered ART after plasma viremia reached its set point. This ART is shown in blue, and reduced the viral load down to undetectable levels. In 2 animals, we interrupted ART and plasma viremia rebounded in the first week. Then we resuppressed, and while the animals were virally suppressed, we administered between 30 million and 100 million CAR T cells. We monitored engraftment of the CAR T cells by qPCR and found in the blood acute expansion of the CD4 CAR vectors at day 7 that was not observed with the CA control CAR vector. Additionally, we observed gene marking of CD4 CAR T cells in the lymph node and gut 6 weeks after adoptive transfer. We followed these animals for almost 2 years and found CAR T cells in the blood transduced with all vectors. These data imply that long-term persistence was based on the CMV specificity rather than on the CD4 CAR activity. When we analyze plasma viral load in this animal with the control vector, virus rebounded on the 3rd week up to 10 to the 4 or 10 to the 5 copies per mil. In the animals with the therapeutic vector, viral rebound was delayed up to 6 weeks, especially in comparison to the rapid viral load in the standard STI. Viral load peaked in one animal and then established a set point around 10 to the 3 copies per mil. In a second group of animals, we used a bicistronic vector expressing either CAR2 or CAR3 and the C46-MAC fusion inhibitor. The CAR2 vectors express 2 intracellular signaling domains, CD28 and the TCR zeta, or 3 intracellular domains, CD28, 41BB, and the TCR zeta. They're also designed to co-express the CD46 fusion inhibitor to protect the CAR T cells from becoming infected. These vectors allowed us to detect expression of the CD4 CAR and to determine the phenotype of the CAR T cells. Cells were stimulated and transduced and then stained with CD28 and CD95. Cells were gated into naive, central memory, and effector memory phenotype. These data show that CAR expression is detected only on the T effector memory cell populations. After adoptive transfer, we followed expression of the CAR T cells in the effector memory population. We observed an expansion of the CAR T cells on day 7 in this population, which subsequently waned over time. Expansion of the CAR T cells was confirmed by qPCR only for the CD4 CAR vectors shown here in blue, but not for the CEA CAR or the CEA CAR control vectors shown in gray. This data suggests that the expansion at this time is due to stimulation through the CD4 CAR vector. We also analyzed activation status of the CAR T cells by expression of CD69. In comparison to bulk CD8 T cells or CD8 positive effector memory cells, the CAR T cells had significantly more CD69 expression, which also peaked on day 7, but was increased throughout. From these data, we concluded that CMV-specific T cells can be expanded and transduced ex vivo to produce therapeutically relevant number of autologous CD4 CAR T cells. After adoptive transfer, CD4 CAR cells peaked at day 7 post-infusion in the blood. Acute expansion is specific to the CD4 CAR T cells and was not observed in the CEA control population. These cells are CD95 positive, CD28 negative, which is consistent with an effector memory phenotype. They are highly activated CD69 positive cells and have reduced expression of PD-1. Persistence of the CAR T cells out nearly 2 years suggests persistence is imparted by the CMV specificity rather than the CD4 CAR vector. Long-term persistence of these cells only limited the control of plasma viral load. Therefore, CAR T cells may require some latency reactivation to increase the susceptibility to cytolytic activity. So, in collaboration with Dr. Partha Chandra and Debashis Mandal, we explored the use of MSCs in HIV. Mesenchymal stem cells are fibroblast-like cells isolated from the bone marrow or adipose tissue, actually from multiple other tissues. Mesenchymal stem cells have multipotent progenitor activity. They can form bone and cartilage in vivo, and they can differentiate in vitro into osteoblasts, chondrocytes, and adipocytes. My experience with MHCs is based on their role in regulating hematopoietic stem and progenitor cells. These cells provide a framework for the hematopoietic microenvironment and to the hematopoietic stem cell niche. They express a variety of cytokines and growth factor and have anti-inflammatory effects in vitro. Just as with hematopoietic stem cells, mesenchymal stem cells interact with T cells and macrophages. These data show 3 different cell populations cultured individually or co-cultured with mesenchymal stem cells. You can see that the mesenchymal stem cell changed the adhesive characteristics of all 3 populations, in that the cells favor interacting with the mesenchymal stem cells rather than forming a clump of cells. So we measured the effect of MSC co-culture or MSC conditioned media on the reactivation of HIV latency. Here in the U1 cell line, the U1 cells were co-cultured with conditioned media from adipose-derived stem cells, and P24 GAG production was measured. So as you can see, in all 4 populations, co-culture with conditioned media from mesenchymal stem cells increased HIV viral reactivation. To address the mechanism of viral reactivation, we transduce U937 cells, the parental cells for the U1 latency model, with an HIV-based lentiviral vector expressing GFP from the HIV LTR promoter. With standard media, there was low levels of GFP expression, But when we co-cultured this population of cells with conditioned media from mesenchymal stem cells, either at 25 or 50%, the frequency and the intensity of GFP positive staining was increased, which indicates that HIV reactivation with this mesenchymal stem cell factor is through the HIV LTR and independent of TAT. To compare MSC reactivation with other LRAs, we stimulated U1 cells with conditioned media or with different HDAC inhibitors. In the left panel, you can see that the conditioned media from ASCs significantly increased viral reactivation. This is P24 GAG production, where the HDAC inhibitors only modestly induced viral reactivation. When we combined the ASC conditioned media with panobinostat, we saw significant increases in viral production. So here's the panobinostat by itself and modest reactivation. The ASC conditioned media alone by itself giving significantly increased viral reactivation, but the combination of the panobinostat and the conditioned media significantly increased viral replication over 100 times higher than the panobinostat alone. In conclusion, during co-culture, monocytes and T cell prefer to interact with and adhere to mesenchymal stem cells. It also affects their mobility and migration, but I didn't discuss this data. Both direct co-culture with MSCs and exposure to conditioned media caused viral reactivation and increased production of HIV GAG P24 from latently infected cells. Conditioned media increased GFP expression from the HIV LTR in a TAT-independent manner, and conditioned media from MSC caused significantly more reactivation of latent HIV than clinically relevant doses of HDAC inhibitors. Additionally, conditioned media synergized with HDAC inhibitors to increase HIV GAG production. So we are exploring the use of MSCs and MSC reactivation factor to enhance the targeting of CAR T cells. And this slide is just to acknowledge the contribution of those that did the work. Nathan Johnson and Chengzheng Wu did most of the CAR T cell work, and Partha Chandra and Debashish collaborated on the mesenchymal stem cells. This work could not have been accomplished without the CMV peptides that we got from MIDI-CAR, and the original CAR project was initiated in collaboration with Dr. Richard Jungens in Boston. We got the antiretroviral drugs and the virus from George Shaw. Additionally, Ann Lean from Baylor provided her peptide stimulation protocol, which we modified to enhance stimulation and proliferation. And of course, the grant support that helped fund these work. So anyways, thank you for your participation, and we look forward to an active discussion.
I think I'm unmuted now. Yeah, okay. Thank you very much, Dr. Braun. Our next talk is titled Modeling the Next Generation of Gene Therapy Approaches in Non-Human Primates. And our speaker is Dr. Hans-Peter Keim. Dr. Keim is the director of the Stem Cell and Gene Therapy Program at the Fred Hutch Cancer Research Center and the chief of the Scientific Unit on Gene Therapy and Regenerative Medicine at the Washington National Primate Research Center. Dr. Keim?
Hello. I would like to thank the organizers for inviting me to present at the 60th anniversary seminar series for the National Primate Research Centers to celebrate 6 decades of biomedical research. My title today is Modeling Next Generation Gene Therapy Approaches in Non-Human Primates. Now, there are obviously many potential diseases and conditions that can be targeted by hematopoietic stem cell gene therapy. You can see the largest populations are people living with HIV or patients with hemoglobinopathies. But overall, there are probably about 60 million people that could benefit from this therapy if it were more accessible. However, so far, less than 1,000 patients have been treated with hematopoietic stem cell gene therapies. We actually, with many of these potential indications also, and have already translated some of these findings that we've learned from in the non-human primates into clinical studies. Now, while there's quite a bit of success with hematopoietic stem cell gene therapies now for genetic diseases, there are a number of limitations currently. In particular, I'd like to address 2. There's currently the requirement for a large amount of vector for editing these hematopoietic cells. Now, this could be, of course, improved by targeting fewer cells, that is, more enriched cell populations. Also, currently, the way we do stem cell gene therapies with the ex vivo approach and ex vivo culture requirements, we do need highly specialized facilities. So this is limited to these facilities. Now, if we could manufacture in a portable device or even do it in vivo, this would be, of course, preferable, since that would be much more portable than the current ex vivo setting. Now, all of the current hematopoietic stem cell gene therapy and gene editing approaches use CD34 cells as target populations. And as we know, most of these CD34 cells are not hematopoietic stem cells and do not contribute to hematopoietic reconstitution. So we waste a lot of reagents and, of course, also increase the potential toxicity and risk for insertional mutagenesis by modifying all CD34 cells. So when Stefan Rodke came to the lab a few years back now, I asked him to look into all the different antibodies available to characterize human hematopoietic stem progenitor cells and test whether they would cross-react with non-human primate cells, and then also test, of course, whether these antibodies would indeed have similar characteristics in non-human primate cell populations as they do in humans. And that's been done, he's done this in colony forming cell assays. And he found 3 distinct populations with 3 conserved markers, the CD34, CD90, and CD45RA. And these populations are shown here. With HSC, those are the CD90 positive populations. Then you can see the MPP for multipotent progenitor cells, the CD90 negative population, and then the lymphomyeloid progenitor population. In blue, those are CD90 negative and then CD45RA positive. So if we could just focus on this CD90 positive population, this is only about 5 to 10% of the overall CD34 cells. This would mean a 10 to 20-fold reduction in cell number, and therefore the 10 to 20-fold reduction in viral vectors or gene editing reagents. Now, to test whether these markers indeed select and identify long-term repopulating cells, we have to do this in a non-human primate study. And again, these studies cannot be done in a mouse because the markers are unique to the human hematopoietic stem cell or the non-human primate hematopoietic stem cell. Now, in non-human primates, we can use these different cell populations and mark them differentially with different fluorochromes, and then infuse them back into a animal that had received myeloablative conditioning. So we can track these populations in the animal, short and long term, and follow the contribution to hematopoiesis. Now, this was one of my most stunning and amazing results. All the repopulation came from this hematopoietic stem cell population, the CD90 positive population up here, early and very persistent and durable, as you can see here. Also very strikingly, all the other lineages came from this CD90 positive population, and this lymphocytes here about day 60 to 90 as expected. Almost nothing from these other two populations. We've also switched the fluorochrome, so marked, labeled this time the CD90 positive population with the M-Cherry fluorochrome, and exactly the same result as you can see here. Only very little few blips with the other colors here with the other populations, and again the same pattern for these other lineages. And it's again very durable here. This is 1500 days after transplantation, and in very high level and very persistent. And the only, in one animal we saw a bit more fluctuation over here, and it was lower because this animal had the lowest cell number overall. We also then, of course, tracked these clones in these animals very carefully. Since we marked them with the fluorochromes, we could sort and analyze these individual cell populations. The different colors here indicate the contribution of the clones to the different lineages. Blue indicates clones that contribute to 5 lineages, for example, granulocytes, cells, B cells, monocytes, and NK cells, and therefore are the very multipotent stem cell populations. And you can see, we see this the blue bars coming on within days after transplantation, of course, increase over time. So we see the CD90 population contributing very, very early and can contribute to all the different lineages over time. Now, this was all gene therapy with a lentiviral vector that we've been using to mark these cells. With lentiviral vectors or retroviral vectors, we will have a pseudo random integration. So we cannot really control. We could hit a proto-oncogene. And this is why many investigators have gone to gene editing and targeted integration with these editing tools. CRISPR-Cas, for example, we can now specifically target a location in the genome and potentially a safe genomic locus or a disease-specific locus. Of course, there's different nucleases that can be used. This just shows you the general principle. We get a double-strand break, and then we get here usually the easiest pathways, non-homologous end joining. If we add a template here, then we can get homology-directed repair, which is much less efficient. And very important for what I'm going to be talking about next for hemoglobinopathies is this microhomology-mediated end joining, which can really cut out up to about 15 base pairs with a simple cut. This is called, again, microhomology-mediated repair. In 5 to 25 base pairs can simply be deleted because of these microhomologies around the cut. And that's, again, important for the fetal hemoglobin reactivation I'm going to be talking about. Again, Sickle cell disease and thalassemia are very common, currently very common targets for gene therapy with lentiviral vectors, but also for gene therapy, gene editing with CRISPR-Cas or other nucleases. And this is, of course, a disease that has a significant burden. There's about 300,000 sickle cell disease patients born with every year around the world, and 60,000 with beta thalassemia. And you can see on this graph in the lower right, the overall life expectancy is still substantially lower when a patient with sickle cell disease, 54 years versus 79 years in the general US population. This just demonstrates how fetal hemoglobin decreases after birth. As you can see here, this is months after conception here. And when you look then after birth here, how it declines, and the sickle cell hemoglobin increases after birth, and will then cause symptoms. And of course, the idea with gene therapy here then is that we will increase the fetal hemoglobin again, and which should then also decrease the sickle cell hemoglobin, and hopefully also ameliorate or get rid of the symptoms from sickle cell disease. There's currently several different approaches. We can either knock out here the fetal hemoglobin inactivator, knock out, block the repressor, or we can down here we can cut out or block or eliminate the binding site for the repressor. And this is what we've done. We've deleted this 13 nucleotide deletion. We've inserted this 13 nucleotide deletion, which is called the hereditary persistence of fetal hemoglobin, which has been shown to increase fetal hemoglobin to levels very close to what would be considered curative. We need probably about 30% of the overall hemoglobin protein to be fetal hemoglobin for patients with sickle cell disease to not have any symptoms and likely be cured. So we've done this again in the non-human primates, just the outline here. But we take the hematopoietic stem cells, enrich them for CD34 or CD90, and then with electroporation and modify the cells using CRISPR-Cas RNPs and knock out this 13 base pair stretch that constitutes the binding site for the BCL11A repressor. We then followed the animals, again, just as before, long after transplantation, as just shown here. We modified the CD90 positive cells only, so we only knocked out the 13 base pairs in the CD90 population, not in the CD90 negative population. Infused both populations into the animal and then followed the animal, short and long term. And this is very striking for us, as you can see here. We see again with the CD90 population and CD34 population, very similar outcomes. What you're looking at here is the percent hemoglobin editing by MiSeq at the gamma globin locus. Again, very consistent. We also see the same for the F cells. Those are the cells now in the blood containing fetal hemoglobin. So very promising. And of course, this has also already been translated into the clinic as well now. Now I'd like to switch to HIV. there are almost 38 million people living with HIV around the world, and about 35 million people have died from AIDS-related illnesses since the start of the epidemic. Now, while antiretroviral drugs have been able to control the virus and thus improve the lives of millions of people living with HIV, we know that these drugs cannot eradicate the virus and thus cannot cure HIV. Now, this all changed, of course, with Timothy Ray Brown, and when he received an allogeneic bone marrow transplant for acute myeloid leukemia in 2007. He received this transplant from a donor who was also resistant to HIV. So that donor had the CCR5 delta 32 deletion, and this prevents the entry of HIV. And then Timothy also stopped his HIV drugs without any viral rebound. And this is how he learned that he was cured in the end and became the Berlin patient. Since then, there have been other transplants without the delta 32 mutation, and those patients all had viral rebound. Those were called the Boston patients. But then finally, in 2019, there was a second patient who also received cells from a donor with a CCR5 delta 32 deletion. That patient was also cured. And now we, in fact, have a third patient as well. Now, of course, this was done in the allogeneic setting, and we knew we could do this with the patient's own cells by simply knocking out the CCR5 receptor in the patient's own cells. And this can be done. If we do this, of course, in the stem cell, then all the cells will be CCR5 negative. And then, of course, all the cells should be protected from HIV infection. Now, we can, of course, use, again, viral vectors or gene editing for the modification to knock out CCR5. We can do this in T cells directly as well. And we can also separately modify the B cells as well if we want to. So we can either modify stem cells to modify all these different lineages, or we can separately modify T cells or B cells. And then, of course, for T cells, for the T cells, we're mostly interested in CAR T cells right now, and for B cells for the production of broadly neutralizing antibodies, or this ECD4-IG fusion inhibitor that has been described by Michael Farsan. We've particularly focused on the stem cell approach, and we've shown very nice results with hematopoietic stem cell derived CAR T cells in this collaboration with the Kitchen and Jerry Zach's lab here, where we really showed a long-term contribution of hematopoietic stem cell derived CAR T cells in non-human primates. This just to show you that these cells persisted and contributed to CAR T cells, also NK cells, so NK cells that had this expressed the CAR construct, and then the CAR construct was also in the B cells detected as well, and in monocytes. So clearly, through the hematopoietic system, we can express the CAR construct in these different lineages, T cells, B cells, and monocytes. We've shown this, that these cells now also traffic to the various reservoirs, to the, as you can see here, to the main reservoirs in the body, the lymphoid germinal centers, the GALT, and the CNS. Now, this is all being done ex vivo, and but really, can we push this in vivo? Because this could not be done, as I mentioned, in any third world country, or where the diseases occur the most, like sub-Saharan Africa. So we sort of back to the same problem as with these transplants. So the question for us was really, could we do this differently? Now, we had previously described this approach using a gene therapy in a box where we do everything in one device, but that still requires a big device. Ultimately, as I mentioned, we'd like to do this in vivo. Then we could simply inject the virus or the lipid or nanoparticle with the editing technology and modify the cells directly in the patient. And that's exactly what we have pursued here with nanoparticles. We've done this with polymer nanoparticles, and have also worked with here with Drew Weissman from UPenn using lipid nanoparticles to deliver these editing reagents to cell populations ex vivo and in vivo. And we've been particularly interested in a collaboration here with Andre Lieber and Andre Lieber's laboratory to use a helper-dependent adenoviral vector system. This vector system, there's all viral genes are removed, so it opens up like 35 kb space for transgenes, has a fiber knob that is actually targeting the CD90 positive, CD46, which is really highly expressed on the CD90 positive cells. And we can combine it with a transposon system, the Sleeping Beauty system, to integrate our payload into the genome, or we can simply deliver in an episomal state the editing nucleases. Here's an example of this in vivo delivery using this helper-dependent adenoviral vector system targeting CD45. What we can include here in this system is a selection step with the MGMT-P140K. So what you can see is we simply mobilize the hematopoietic stem cells in these animals and then select afterwards with a very low dose of BCNU. And when we select for these now modified hematopoietic stem cells, you can see very nicely with just 4 selection steps, we can get the modification levels up to about 30% here, so clearly in a therapeutic range. Now, if there's a disease background, this is in, for example, in mice, in thalassaemic mice, we can even get a higher baseline modification here, about 20%. And again, with a simple in vivo selection approach here, titrate this up to the required levels in vivo. Now, of course, importantly, we want to test this in non-human primates to really see, can we translate this potentially into a human system? And here you can see, we've seen also very efficient modification in non-human primates. Again, very low initially, but then with these selection steps, we can get up to higher than 90% of red blood cells that were gamma globin positive. So really contain the fetal hemoglobin. That, of course, would correct the sickle cell phenotype. So we're very excited with these findings, again, in the non-human primate model. That was critically important for us to show, first of all, that it's doable, so it is feasible in terms of toxicity and in terms of course, efficacy. Okay, so we hope that we will be in the future, we will be able to push this, the efforts to in vivo rather than doing this laborious ex vivo approach in these specialized facilities. There's also been significant effort by the Bill and Melinda Gates Foundation and NIH to support this in vivo gene therapy approach. And would like to end with this quote from Francis Collins: "Such treatments that happened entirely within the body, known as in vivo treatments, would be a major step forward from current treatments." And with that, I'd like to finish and conclude, I'd really like to thank my team. We've got a spectacular team, and especially the non-human primate team at our primate center, and of course all the staff at our non-human primate center at the University of Washington. It's been wonderful. It's been wonderful to work with everyone within our non-human primate center. Thank you very much.
Thank you very much, Dr. Keem. Our third presentation is titled Precision Medicine for Neurodegenerative Disorders. And this will be presented by Dr. Marina Emborg. Dr. Emborg is a professor of medical physics at the University of Wisconsin and the director of the preclinical Parkinson's research program at the Wisconsin National Primate Research Center. Dr. Emborg?
Hello, everybody. I'm honored to celebrate with you the 60th anniversary of the National Primate Research Centers. I'm proud to be part of the Wisconsin NPRC, and as such, to be part of the NPRC network. Non-human primate research has played a critical role advancing novel therapies for neurodegenerative diseases. Precision medicine is now the new frontier. Today, I will share with you my team's work developing precision medicine approaches for these diseases. Precision medicine is envisioned as a personalized strategy to deliver a targeted, optimal therapy to the right patient at the right time. For these approaches to reach their target goal for neurodegeneration, it's key to first take a step back and understand the disease target. The two most common neurodegenerative disorders are Parkinson's and Alzheimer's disease. Both are age-related disorders. Parkinson's is classically defined as a movement disorder associated to the loss of dopamine-producing neurons in the area of the brain termed substantia nigra. Alzheimer's is typically classified as a dementia, with patients having memory loss and neurodegeneration in the hippocampus. But these descriptions are an oversimplification of the patient's problems. Let's think about Parkinson's disease as an example. In addition to the typical motor symptoms such as slowness of movement and tremor, PD patients present a myriad of non-motor symptoms such as depression, anxiety, and constipation. Most importantly, symptoms vary between patients and they change over time with disease progression. This range of symptoms is because over time, PD affects multiple central and peripheral nervous system areas beyond the substantia nigra. It is proposed that the progression of neurodegenerative disorders is linked to the progressive accumulation of proteins across the neural axis, mainly alpha-synuclein in Parkinson's disease forming part of the Lewy bodies, and beta-amyloid and tau in Alzheimer's disease, forming plaques and tangles respectively. These proteins are also seen in other disorders, like, for example, frontotemporal dementia may have accumulation of tau. Regardless, each of these proteins seems to follow a different neuroanatomical pattern of progression over time. Thus, in addition to the pathology, it is critical to consider the timeline of the disease. The disease stage when planning therapeutic interventions for these disorders is also critical to keep in mind when we're testing therapies in animal models. If we want to prevent disease progression, early diagnosis and treatment are needed in order to have a greater chance to have meaningful results, while restorative approaches are suited for advanced stages. Let's first consider precision restorative approaches. Cell replacement strategies are an example of a restorative approach. My team, in collaboration with Dr. Shang, has developed a cell-based personalized approach for Parkinson's disease that we have tested in rhesus macaques with hemiparkinsonian induced by administration of the neurotoxin MPTV. As if we were working with patients, my team collected skin fibroblasts from a monkey and gave the cells to the Shang lab. They first generated induced pluripotent stem cells and then differentiated the iPSCs into midbrain dopaminergic neurons, which he then gave back to us for transplantation into the NPTP-treated putamen nucleus. My lab used real-time intraoperative MRI methods for delivering the cells into the brain. Using a battery of behavioral and imaging methods, we compared a group of animals that received autologous grafts from the same subject versus a group that received allogeneic grafts from a different subject. Dr. Tao prepared one cell line per autologous animal and differentiated them into midbrain dopaminergic neurons. None of the monkeys in the study were immunosuppressed. Using PET imaging, we found that after NPTP, all monkeys had a unilateral loss of dopamine activity in the brain. That persisted in the animals that had allogeneic grafts. In contrast, the autologous grafts recovered dopamine activity where it was lost, which is, you can see it here with a white arrow. When we look into the monkeys' brains 2 years after grafting, we saw that the allogenic neurons were present, but the grafts were smaller, more compact, and the neurons had smaller extensions, while the autologous grafts integrated and extended their neurites into the host parenchyma. The cell survives with minimal immunological reaction from the host, which most probably facilitated the graft integration. Most importantly, we saw an improvement in the Parkinsonian motor and depressive behaviors of the animals with autologous grafts. And the amount of improvement correlated both with the amount of dopamine activity measured by positron emission tomography signal and with the number of Th-positive dopamine-producing neurons in the graft. Based on our results, we can predict how many cells are needed to improve Parkinson's disease symptoms in humans. The ratio of rhesus to human glutamin, the target area for dopamine cell replacement, is approximately 1 to 4. Thus, if 50% improvement in rhesus is observed with approximately 70,000 surviving dopamine cells, a similar effect in humans will require 300,000 dopamine neurons located precisely in the target. This approach could be further personalized based on the patient's brain imaging data to define graft placements. And we hope that in the future, the cells could also be adjusted by genetic condition, as today we have the tools to genetically modify the cells. Speaking about genetics, now let's consider novel approaches to protect neurons based on precision medicine. As I mentioned before, early diagnosis or better yet prediction of who could have the disease would increase the chances for success of a neuroprotective strategy. The problem is that the cause of Parkinson's and Alzheimer's disease is unknown. Yet, gene mutations have been linked to both diseases. Thus, early identification of mutation carriers may help identify potential candidates for early neuroprotective interventions. In PD, a number of autosomal dominant and recessive mutations had been identified. Of those, LRRK2 G2019S mutation is the most frequent. But how frequent is the most frequent PD mutation? Well, to keep it in context, let's think about this. PD affects 1% of the population over 65 years old. Of those 1%, 10 to 20% are familial, and approximately of those 2 to 40% carry the mutation dependent on the anestheticity. Thus, it is critical to think carefully the gene target and the model before devoting resources, because the development of the non-human primates genetic models is something that is difficult to achieve and have signs, symptoms, and pathology that matches. This is something that Dr. Schmid and I spent a lot of time thinking about it. Just to think about this a little bit, how we can make monkeys with mutations. Well, the monkeys can carry naturally occurring mutations, which can be identified by genome sequencing. For example, at WNPRC, we have identified a family of rhesus carriers of a tau mutation linked to frontotemporal dementia. Alternatively, the mutation can be introduced to somatic cells in adult monkeys, like Dr. McBride described a few weeks ago regarding the intracerebral injections of the virus vector to express the protein Huntingtin. They can also be introduced into the germline to generate transgenic or genome-edited monkeys. Regardless of the method to have mutation carriers, it is important to consider the monkey species. Each presents advantages and disadvantages that need to be considered. For example, compared to rhesus macaques, marmosets reach adulthood faster and have a shorter lifespan, which is advantageous for aging studies. Marmosets are also very fertile and frequently have birth of twins or triplets, which is great if you are planning to expand a germline modified subject. But we need to think in genetic studies, the target gene and the protein in the chosen studies. With respect to and especially with respect to the neurodegenerative disorders, alpha-synuclein in common marmoset has a naturally occurring threonine in the amino acid 53, which makes marmosets not an ideal species to study this mutation as in Parkinson's disease, as T53 is associated with a form of familial Parkinson's disease. In addition, marmosets, like rats, but not humans and macaques, do not express the tau 3R isoform, which is a problem if you want to study tauopathies. Let's go back to thinking about the LRRK2 G2019S mutation. This means that there is a G6055A point mutation, which results in a glutathione to serine switch at amino acid position 2019 in the kinase domain of the LRRK2 protein. That small one point mutation makes the kinase more active. We wonder if we could apply CRISPR-Cas9 to modify the gene, that's the protein, by using a molecular scissor to cause a DNA break and then provide a single-stranded oligodeoxynucleotide donor template. We could or introduce in a specific nucleotide sequence that change or do a homology-directed repair, which could be used to model or to create a therapeutic strategy. To test this hypothesis, we use common marmoset monkeys stem cells, because we hope to use marmosets as a platform for testing the approach in the future, as the LRRK2 kinase domain is highly conserved compared to humans. The LRRK2 G2090S mutation has not been identified in marmosets, so we first chose to introduce the mutation in the cells, which is something that Scott Milea, former student in my lab, performed. And you can see the G6065 with the green arrow, you can see that point mutation that he introduced. When he analyzed the cells, what he found was that similar to human cell cultures, the LRRK2 G2019S mutated marmoset stem cells have increased kinase activity compared to wild-type cells. Furthermore, when he analyzed the structure of these cells, he found that also like human cell cultures, the dopaminergic neurons differentiated from these mutated stem cells had less number of neurites and branches compared to wild-type cells. So in the LRRK2 G2019S point mutation, the kinase domain is more active, we wonder if we could apply CRISPR-Cas9 editing to modify the gene, thus the protein, and decrease the amount of activity. So Scott, that was another thing that he did. He used CRISPR-Cas9 again, and he used these molecular scissors to cut this part of the DNA to truncate the protein. What he found was that he could decrease the kinase expression. And thinking about the chemical translation, what he found is that the cells with less active LARC2 have a similar morphology than wild type, which was very interesting a strategy to, if you have the mutation, that you could decrease the amount of kinase activity. Now, this was done in culture, so our approach is not so great if you are thinking to do it in a whole organism. So we started thinking, how can we deliver the genomic editing material to whole organisms? So for that, to answer that question, we teamed up with Doctors Gong, Saha, Levine, and Roy to test nanoparticles delivered by Dr. Gong and bring this to bring this CRISPR material into the brain. Our multiple PI team is part of the Somatic Cell Genomic Editing Consortium. We have demonstrated the efficacy of the approach in TB tomato reporter mice. When the nanocapsules are effective, they induce a deletion in the stop codon and TD tomato is expressed, which you can see here in the coronal vein section where the edited area is red. Intracerebral injection of nanoparticles preferentially edited neurons and also a few GFAP positive astrocytes. We quantify the different formulations because we can decorate these nanoparticles to make it more attracted to certain neurons, or at least that was our hypothesis. But when we found this, it didn't matter which type of formulation we use. All of them had more or less a 20% editing efficiency in the injected area. This 1 microliter injections approximately edited 1 millimeter the area, square millimeter area, and extended approximately 2 millimeters across the coronal plane. Our next challenge now is to demonstrate in vivo editing, especially in a clinical relevant target in non-human primates. For this phase, we aim to modify the amyloid precursor protein, aiming to produce a truncated protein that has less chances of accumulating in the brain and form plaques. For this project, we are working with the California NPRC team, which is the large animal testing center for the stem cell genomic editing consortium. To conclude, neurodegenerative disorders are complex. They vary between patients and change with disease progression. And in that context, the selection of the experimental paradigm and animal model, including the species, is critical. Mutated non-human primates, either by genomic editing or by identification through colony genotyping, present unique opportunities for precision medicine approaches. And there is a number of novel therapies that are being developed for neurodegenerative disorders that can be applied for precision medicine, from cell-based therapies to genomic editing. Ultimately, successful clinical translation of precision medicine approaches will require factoring in the target, efficacy, the safety of the therapy, and delivery methods, for which I believe non-human primate research will be a critical clinical step. And with that said, I want to thank my amazing team because this is possible because of their hard work and dedication. And also I want to, I'm very grateful for my amazing group of collaborators I try to highlight throughout my talk. But I want to say a special thank you to our amazing vets and vet techs and animal caretakers at the NPRC. As well, the other behind-the-scene wonderful people that provide invaluable support to make this research happen. Many, many thanks for your attention and happy 60th birthday NPRCs, and wishing you another happy 60 more. Thank you.
Thank Thank you so much, Dr. Emborg. And thanks to all of our speakers for really fantastic talks. It's a super exciting field. So I'd like to open up the floor for questions for our speakers. And I'm not quite sure if we can see our speakers. I see Marina, there you are. Hi. Great. And Steve, Hans Peter, welcome. Great to see you all. So we'll give it a little time for some questions from the audience, but perhaps to get us started, I can ask a question or two. I'm having trouble moving my screen here. Yeah, here we go. So I'll go ahead and just ask a couple of questions to get us started. So Steve, you mentioned something about reactivation, viral reactivation. Can you comment a little bit about this MSC reactivation factor, perhaps?
Well, I was a little bit surprised in even finding that the mesenchymal stem cells were reactivating latency, but I mean, thinking about it, they secrete a multitude of cytokines and growth factors, any one of which could probably, well, not anyone, but which with the potential for reactivation. So we haven't identified the factor, but what we have done is we know that it's a heat labile factor because we can destroy the activity by heat inactivation. And we also know that it's signaling through PI3 kinase. So it's some sort of factor. And we also know that if we activate MSCs through TLR3, that we can upregulate the reactivation factor, but not through TLR7. So there's some sort of specific reactivation or activation of the MSCs that will induce the factor. So, you know, we're keenly interested in identifying the factor. And, you know, in terms of personalized medicine, I think a cytokine would be more, how would you say, clinically relevant than having to isolate personalized MSCs from an individual to get reactivation. So if we can identify the factor, it would increase its clinical utilization.
Mm-hmm. Great, thank you. There's a follow-up question here regarding if there's evidence that the MSCs can reactivate latent SIV in vivo.
We have not been able to do that experiment yet. That's a funding issue. But we do know that rhesus MSCs will reactivate latent SHIV from T cells and infected monocyte macrophages. So we have done the experiment in monkeys or in vitro, but not in vivo.
Thank you. There's a question here for Dr. Emborg. An attendee would like to know whether the transplanted animals showed any evidence of graft-induced dyskinesias, as has been described in the past with human transplant recipients.
Thank you for the question. We looked for it. We did not do a dopamine challenge, but we looked for spontaneous and we did not see it. We followed the animals for 2 years. And
I'm sorry, Marina, I'm having a little trouble hearing you. Is that just me or?
Okay, let's see. Hello, can you hear me now?
Oh, that's better. Yeah, that's better.
Okay, so I'm using my other microphone. So what I was saying is we have not found dyskinesia to follow the animals for 2 years, and we did not identify a spontaneous occurring dyskinesia.
Okay, thank you. And let's see, I would be interested, another question for you, Dr. Emborg, is I wondered if you could talk a little bit more about this frontotemporal dementia. You mentioned it sort of briefly, I think, but I'd be eager to hear a little bit more about that.
Yes, that is my new favorite new project. Um, in collaboration with Jeff Rogers at Baylor and Rick Ken, we have been genotyping all the animals in our colony and at the Primate Center here in Wisconsin. And we found one animal originally that has a tau mutation, the R406W mutation, and has been linked in human patients to frontotemporal dementia. And so after that, we have identified a sibling and then three other kids that the female rhesus had. So at this moment, we have 5 animals that are between 18 years old to 1 year old with this mutation. And this is very exciting because they have changes in MRI and in behavior. Especially, they have changes in mood, some changes in gait that we have seen in Parkinson's in patients and very similar gait problems, and also some issues with cognitive, uh, um, resolving cognitive tests. So we're very excited about this project.
Yeah, that's super interesting. Yeah. To your knowledge, is that the first time this spontaneous type of dementia is found in this population, or yeah?
This is, as far as I know, this is the first time. So
Yeah, that's really, really fascinating. Yeah, yeah. Yeah, I wonder if it's just because we haven't looked before, it's
well, this animal is 18 years. The older animal is 18 years old, so obviously you have to look for it. I think it's really cool that Dr. Dr. Rogers is helping us all identify these animals across different primate centers. And he can highlight different mutations. What is interesting of this specific mutation is autosomal dominant. So you can have a heterozygous animal and you can have a phenotype, which is harder with other mutations.
That's really interesting. Thank you. Thank you. I think there's another question here for you, Dr. Emborg. The marmoset AT substitution at AA53 and alpha-synuclein, is there a compensatory mutation in marmoset alpha-synuclein that allows normal protein activity?
That's an excellent question. As far as I know, there is not. This is across all the species of vertebrates, with exception of macaques, baboons, apes, and humans. All the rest of the vertebrates have the 53T naturally occurring. So, yeah.
It looks like you're pretty popular here on the chat, Marina. So there's another question for you. So, hi, Marina, great talk. A technical question. Can lipid nanoparticles be delivered to the NHP brain using convection-enhanced delivery to improve spread, similar to what is done with viral vectors? Wasn't sure if it compromised their integrity.
So first of all, what we use are not lipid nanoparticles, just to make sure. These are ribonucleoproteins. It's a current ribonucleoprotein with all these different pegged ligands. And then it can be decorated with rabies virus glycoprotein or cell-penetrating protein. So in this particular case, yes, we are exploring the convection-enhanced delivery. Lipid nanoparticles, although I have not used, other colleagues have used with convection-enhanced delivery,
not for ribonucleic proteins, but for delivering other compounds. And if you refer to the integrity of the lipid nanoparticle, my understanding is the integrity is maintained.
Thank you. And one more, don't rest. So thanks for the interesting talk. Did the transplanted animals show any evidence for graft-induced disc? Oh, we've already asked that.
Yeah.
Whoops, we already asked that.
I think
Okay,
there's one
we'll
for
skip that one.
Steve.
Yeah, and then here's one. Yeah, here's one for Hans Peter. Dr. Keem? Yeah, here we are. In proposing in vivo gene delivery, how would you propose targeting all the HSCs in different bone marrow niches?
Oh, very good question. So we'll probably not be able to target all of them, but what we've also incorporated in our vector system is a selection mechanism. So we can actually select for the modified cells. So we don't need to target all of them, but we select for a large proportion of them after we've modified them. That's the trick.
Great. Any additional questions, topics the speakers want to talk about, perhaps? Okay. If not, I'd say we can declare a victory. Thank you so very much. It was really wonderful. Really enjoyed the talks and really appreciate the great conversation. Thank you.
Great.
Thank
Bye,
you.
everyone.
Oh,
Uh,
pleasure.
Thanks everyone for coming. Take care.
Take care.
Bye-bye.